My name is Sue Wessler. I'm a professor of genetics at the University of California, Riverside, and my lab studies transposable elements. The title of these two presentations are the dynamic genome. In the first talk, I introduced transposable elements by describing their discovery by Barbara McClintock, how they move, and how that discovery over the years was recognized as a major revolution in biology as it became appreciated that transposable elements are the major component of most of the genomes of higher eukaryotes. In this talk, I'm going to go into detail about how my lab studies the evolutionary impact of transposable elements on genomes and how we developed strategies to identify elements that have an impact on transposable elements. I've divided this talk into three parts. In the first part, I talk about the transition from genetic approaches to genomic approaches in order to identify elements that, in fact, impact genome evolution. The elements that were discovered in my lab are called MITEs, and I'll tell you about that discovery in the second part of this talk. And in the final part of the talk, I will tell you about how MITEs are able to increase their copy number in the genome without harming the host significantly. So to review the first part, we talked about the genetic analysis of transposable elements, how genetic analysis led to the discovery of transposable elements. And I use this spotted corn kernel as an example to tell you really how powerful the genetic analysis was. So when you see a spotted corn kernel like this, we know, the geneticist knows, that the reason that kernel is spotted is because there are active transposable elements. There are transposable, that is the spots reflect the movement of transposable elements. The other thing that the genetics tells us is exactly where in the genome that active transposable element is. So for example, here when we're looking at spotted corn kernels, we know that there is an active transposable element. In other words, one that is capable of moving in a gene responsible for kernel pigmentation. The other thing that the genetics tells us is the type of element that's there. And I described at the beginning of the talk, at the first talk, the difference between autonomous elements, that is elements that encode transposase, and non-autonomous elements. Those are the elements that do not make transposase, but are able to move if there's an autonomous element in the genome. McClintock and others were able to deduce this just by looking at the behavior of transposable elements in crosses. Now that's the good news about the genetic analysis. Unfortunately, the genetic analysis is limited in its scope. And that is that by its very nature, genetics depends on the analysis of mutant alleles. And so the transposable elements that were being studied were transposable elements causing mutations. They were mutagenic elements. Now, because these elements cause mutations, there aren't many copies of them in the genome. So I mentioned in the first talk that genomes are up to 50 to 80% of the genome sequence is derived from transposable elements. However, the elements that cause mutations are not those elements. And you can understand that an element that causes a mutation is eventually, if its copy number increases too high, will kill the host. So these are a special class of transposable elements that cause mutations. And as such, these elements really have a minimal impact on genome evolution. They're bad. They're really bad. So McClintock, as I also described in the first talk, not only discovered transposable elements, but she hypothesized that they were also tools that diversify organisms. And to review, she hypothesized that transposable elements that are in the genome do not move around frequently, that there are conditions, that changes in climate, for example, that could activate transposable elements, that this activation would generate genetic diversity in the population and by increasing the frequency of mutation, and that some of these transposable element mutations may be adaptive. I will come back to this scenario at the end of the talk when I show you how the elements that we've identified in plant genomes fit this scenario very, very nicely. So to review, I described in the last talk the two general classes of transposable elements in the genome. The first class, which is called class 2, are DNA transposons. These are the elements that were discovered by McClintock genetically. We know these elements have a typical structure of terminal inverted repeats, that they encode a single protein that's necessary for the movement of the element, and that's transposase. The other class of elements, which I'm not going to go into extensively in this talk, and nor did I in the last talk, are called retrotransposons. These are elements that encode reverse transcriptase, and they move through an RNA intermediate, again by mechanisms that were described in the last talk. I also want to reintroduce you to transposable element families, because we're going to revisit families in this talk. That transposable element families contain autonomous elements, that is the elements that encode transposase, and non-autonomous elements. These are elements that don't encode transposase but are able to move utilizing the transposase that's encoded by the autonomous element. Okay. Something else that I discussed in the first talk is just how prevalent transposable elements are in genomes. So this was a human gene, and where I showed you the exons that are in the gene. And this is really a pretty typical gene. And what we find is that in the non-coding regions, there are many, many, many transposable elements. That some human genes have over 100 transposable elements in their introns. Now, the element that I'm referring to, most of these elements, in fact, in the human genome are called ALU. They're a class 2 retrotransposon that are present at an astonishing copy number of over 1 million copies of this element. It's almost 10% of the human genome. Now, one of the things, if we're interested in the evolutionary impact of a particular transposable element, one question we could ask is, so for example, if we pick out one of these elements, we could ask, what happened when it inserted? Did it change the expression of the gene? These are the questions, if we're interested in the evolutionary impact, these are the questions that we'd like to be able to address. Unfortunately, we can't do that with the human elements, and with most of the human elements. And that is that the insertions may have changed gene expression, but we have no way to address that now. And the reason is, first of all, in the human population, virtually all of us, 99.99% of us, have these insertions, have exactly these insertions. That's because these elements moved millions of years ago. So what that means is if we want to know how did the insertion of a particular element change the expression of a gene, if at all, we're too late. So what we want to do is identify a group of organisms where these high copy number elements are actively transposing. And I'm going to talk about that strategy. That's exactly what this talk is about. So here's our strategy for analyzing the impact of transposable elements on genome evolution. And this is a figure that from the previous talk, which is a sort of typical region of a genome, grass genome, and this is from the barley genome. And the blue boxes are genes, and the triangles are transposable elements. So in barley, about 85% of the genome is derived from transposable elements. So the strategy that we would like to do to identify evolutionarily relevant transposons is to find a species that is in the midst of genome expansion. So where these high copy number elements are moving, are increasing their copy number. And we would then go ahead and identify and isolate an active element. So this is not one of the mutagens that was identified by the geneticists, but in fact, these are the high copy number elements that are now increasing in copy number. Okay? We would then ask the question, how is this element able to increase its copy number so extensively without harming the host? What are its strategies for success? And success in this case is defined by being able to increase your copy number without killing or harming the host, and possibly even by benefiting the host in some way. And we're going to address all those issues in this talk. So in the first talk, we talked about the discovery of transposable elements in maize. Well, maize is a member of a larger group of organisms, a member of the grass. It's a grass. These are the most important organisms for human health, for the human diet. More calories come from members of the grass clade than any other group of organisms on this planet. We're familiar with maize. The other members of the grass clade is rice, which actually is the most important source of human calories. Sorghum, which is also a very important crop plant, especially in Africa. And finally, barley. Another member of this family, which I'm not showing, is wheat. So what you would notice here, those numbers, are the size of the genome. The maize genome is about the same size as the human genome at 2,500 megabase pairs. The rice genome is much, much smaller. It's almost 10-fold smaller. And what's remarkable is that here are these plants that are so incredibly similar, yet their genome size differs dramatically by more than 10-fold. So these organisms diverged from a common ancestor only about 70 million years ago. And the main reason for this difference in genome size is this dramatic amplification, expansion of transposable elements. And this slide helps explain, in part, how that can happen. So these organisms, the grasses, in fact have about the same gene number. They have about 30,000 genes, give or take a few thousand. And so the genomes of these organisms are largely synchronous. The genes are mostly in the same order. So in rice, you could see with the smallest genome, I've shown 3 genes there in pink, yellow, and blue, that are pretty close together. In maize, those genes are further apart. And in rice, those genes are in barley, those genes are even further apart. So what's happening here is transposable elements, which are the squares and circles and ellipses in between, transposable elements are inserting massively between the genes and expanding the genome. Okay? So this is largely responsible for the difference in genome size. And it's the safe havens that transposable elements can go without harming the host. So I want to show you a little bit, show this at a higher resolution. To show you what elements are involved. So I introduced you before that there were 2 types of transposable elements. There were class 1 elements, which are retrotransposons, and class 2 elements, which are DNA transposons. The retrotransposons, which are generally these big elements that make RNA copies. That RNA copy is then made into double-stranded DNA. The double-stranded DNA can insert back into the genome. It almost makes copies like a printing press, like an old-fashioned mimeograph machine, which most of you probably never experienced. So what you see here is that the huge blocks, could be hundreds of kb, that separate some genes in the grass genome are largely retrotransposons that are inserted into each other, literally driving the genes apart. So as we say, it's almost like genes sitting in a sea of transposable elements. Now, these are not the elements that we're going to talk about today. Instead, we're going to talk about elements that I think are probably more involved in diversifying the genome. And that is, so what I've done here is I've blown up the area of one gene and broken it up into its exons and introns. And I've shown you that sitting in plant genes, much like the ALU elements that are inserted in human genes, are little elements, little transposable elements called mites. These are DNA transposons. They're non-autonomous elements, and in the next few slides I'm going to tell you a little bit more about mites because they were discovered in my lab at least 2 decades ago. So the way the first mite was discovered was it was an insertion sitting in a mutant gene. And this is the work of several people in my lab, especially Tom Bureau and Rita Varagona. What you see here is a maize gene, and sitting in it is a little DNA transposon disrupting the gene, causing a mutation. When Tom Bureau isolated this transposon, Like I said, this is back in the early 90s. When he isolated the transposon, he took the sequence and compared it to several other maize genes and other plant genes that had been deposited into databases. This is before you did blast searches. This is in the early 90s when there were some wild-type genes that had been sequenced. And what he found that, in fact, sequences like this little element were present in many of these other wild-type genes. And I'm showing that here. So what you see is here are some wild-type genes that were revealed by this computer search. And so, for example, this gene has an insertion in an intron. So these are normal genes. So these insertions are in non-coding regions. They're not affecting the expression of the gene. This one is in the 5' promoter region, and the one at the end here is in the 3' region. So he discovered these elements. They're called, the MITE stand for miniature inverted repeat transposable elements. What's similar about these elements is their structure. Their sequence may not be similar and is not similar when you go from organism to organism. But these are the most predominant transposable element type associated with the genes of plants. Okay. So let me tell you where mites fit in a transposable element family. So I told you before about autonomous elements. I've told you about non-autonomous elements. Mites are non-autonomous elements, okay? But they have no coding capacity. So in this case, I've shown them, they look like they're a deletion derivative of the autonomous element, but have none of the coding sequence of the transposase. That's one possibility. The other thing, first of all, mites are very, very small, very short, and they can attain very high copy numbers. So whereas most non-autonomous elements in the genome, there may be 5 or 10 of them, maybe up to 50, there could be thousands of mites, okay? I'm going to talk more about that in a bit. Here's an example of mites that look like an autonomous element that's in the genome. Its terminal inverted repeats, its ends are very, very similar. So we can look at this and say, this autonomous element must move these mites. And we've done lots of experiments over the year to validate that. There are other mites in the genome that don't look like any other element that's in the genome except for the ends, except for the terminal inverted repeats. And I think you might remember from the first talk that the terminal inverted repeats are critical because that's where the transposase binds and facilitates the transposition of the element. So mites are not short, miniature inverted repeat transposable elements. I won't say that anymore. I'll just say mites from now on. They're short elements. They can attain very, very, very high copy numbers, unlike most other DNA transposons. And that's what's relevant here, that it's the high copy number elements that I will argue are the ones that have an impact in diversifying genomes, not the lower copy number elements that, as McClintock and others had discovered, cause mutations. So fortunately, mites are not restricted to plant genomes. And I say fortunately because a lot of my work over the years was funded by the National Institutes of Health. And if you're working on a plant system, it's nice to be able to say that what you find in this plant will be relevant to human health. We all try to say that. So in this case, this is the composition, the transposable element composition of a mosquito genome, Aedes aegypti. And it turns out that about 16% of this genome is derived from mites, from transposable elements. So what I would say in my grant application is by understanding how mites move and how they increase their copy number in a plant genome, we can extrapolate. We can use that information to understand how they expand in animal genomes. And there are mites in zebrafish, in most higher eukaryotes, but none of them to date have been shown to be moving. So the other thing about mites that's really is relevant is that they are preferentially in genic regions. Now remember, again from the first talk, we said that a very small percentage of the genomes of plants are genic. So it may just be like 10 or 20% are where genes are, but mites preferentially go into genic regions. And we'll talk a little bit later about that preference. So we have this situation here where we have these very high copy numbers. So we have elements that expand, that increase their copy number, and they go into genes, but they're not killing the host. So how do they do that? So not only do they not kill the host, but they seem to be beneficial. So what I've shown here are two examples of wild-type genes. In the first one, we see the red exons, and what I'm showing is that transcription, the sequences that, that initiate transcription are actually derived from the MITE sequence that's in the promoter region. Similarly, there are other MITE examples of wild-type genes that have mites that in fact carry the sequences for transcription termination. So mites carry, in some cases, regulatory sequences that are used by genes. Mites also contribute to allelic diversity. So what I've shown here is the same gene, or alleles of the same gene, one with a mite and one without a mite. So here would be a wonderful example to be able to say, okay, I have a gene without a mite, I have one with a mite. How do they differ in expression? And that may be able to tell us what is the impact of insertion of that mite. Unfortunately, when we look at a database and we harvest all of these related sequences and genes, it turns out that these genes don't just differ, the alleles don't just differ by the presence or absence of the mite sequence. They have many other single nucleotide polymorphisms, indels, that differentiate them. And this indicates that this insertion happened a very long time ago. So these are essentially dead elements. And really, if we can't tell by comparing the expression of these two genes what the impact of the mite was, because there's lots of other differences between those genes. So as I said, these are old insertions. Okay? So what we need are active mites in order to understand how they actually, and we need to catch them in the act of increasing their copy number. So what we need is a situation like this, and I'm going to come back to this later, and that is two genes that differ only in the presence or absence of the mite. Then we could compare the two genes and say, okay, if this one, for example, is expressed in the roots and the leaves, and this one is just expressed in the roots, we could say that the mite sequences allowed this gene to be expressed in a different tissue. It diversified its expression. Okay? So we want alleles that only differ by the presence or absence of the mite. All right. So now I'm going to, the next part of the talk is that quest, that search for active mites. And so what I'm showing here is a phylogenetic tree, and it's called a star phylogeny. So the way we interpret these, so what's done is you take all of the mite sequences that are in a genome, you put it into a computer program, and it generates a tree that tells you how these sequences are related to each other. So this is what you see, what this tells us, the story that this tree tells us, is sometime long, long ago, there was a single element or a couple of related elements. They increased their copy number. They were all identical. They increased their copy number. And then somehow transposition stopped, and over the thousands or millions of years, these sequences drifted. They accumulated mutations, and now you see this star phylogeny. So that's the story that this tree tells us, and that's a typical mite family tree. Okay, and as I said, it tells us that mites amplify rapidly from one or a few nearly identical copies. Okay. So if we look at a typical, the genome of a higher plant or animal, what we see are lots of bursts. And for convenience, I'm showing the same tree that I've cut and pasted, but in fact, each of these trees should be different because it's a different mite that started out as one copy and then increased their copy number. So forgive me, because these trees are not easy to draw. So you see, there's just, genomes are filled with mites, tens of thousands of them, that all started from a few elements and bursted, but over evolutionary time. Okay. So in order to understand how those elements amplified without killing the host, we need to, as I said before, catch a mite in the act of bursting. And that's this central region, this red circle here, where the element is rapidly amplifying. And that's what the rest of this talk is about. So in order to identify active mites, my lab had to switch directions. And I think this happens frequently, that sometimes the organism you work on isn't ideal for the questions that you want to address. And so this was from the first talk. I showed you this is the first maize genetics group, or the maize genetics group, R.A. Emerson's group at Cornell. And there's a picture of Barbara McClintock at the end. And here's the spotted kernels that she used to discover transposable elements. And I mentioned that this is a shed in the Cornell plantation. And this picture was taken in 1929. Well, what we did in 2002 is we got a new group of researchers together involving Susan McCouch at Cornell, Sean Eddy, who at that time was at WashU, Zhurong Bao, many other people, and we took a picture in front of the same shed in the Cornell plantations. And so this is our collaborative group that focused on identifying active mites. And to do that, we had to switch organisms, and we switched to rice. And I'll tell you in a second why we did that. I think I mentioned that maize has this really, really large genome of 2,500 megabase pairs, that the rice gene, that it's about the same size as the human genome, very dynamic, very complex. The rice genome is significantly smaller, almost about 6-fold smaller. And it is of this group of grasses that I talked about before. It has the smallest genome of the cereal grasses, 350 megabases. And for that reason, and plus because it's so important to human health, it was the first grass genome that was completely sequenced. And for us to do this project, we required the complete genomic sequence in order to identify the active mite. We weren't going to use a genetic approach, we were going to use a computational approach, and that's what I'm going to talk about. So here's the strategy. We had the complete genome sequence of rice, and when I say we, the person mainly responsible for this, two people, Zhurong Bao, who was a graduate student in the lab of Sean Eddy, who, as I said, was at WashU at that time, and Ning Jiang, who was a graduate student in my lab. Zhurong Bao had devised a computer program called Recon. What this program does is it takes, so what we're looking for, we're trying to find an element, a mite, in the genome sequence that has the features of an active transposon. What are those features? First of all, a transposable element will have many copies. So we're looking for something that has multiple copies, but we're looking for something where those copies were generated very recently. So those copies, remember, we're looking at the red region of that phylogenetic tree. Those copies should be identical or nearly identical. Because when an element duplicates, the two copies are identical. And over time, they drift, and that's what the star phylogeny is there. So what was done is we took the rice genome sequence, compared it to itself, and in that way identified high copy number repeats. Okay? And about 3,000 repeats were identified. These could be genes, these could be transposable elements. Now then the human being has to come in. Well, the human being came in devising the Recon protocol. Okay? But what you have to do, and this is done by Ning Jiang, she manually searched each of these 3,000 families to try to find a sequence that looked like a mite. And she found one, obviously, or I wouldn't be talking about it now. Okay, so what she found was a family that had 51 nearly identical copies in the sequence genome, which is called Nippon-bari. That's the name of the strain. There were 51 copies in this genome, and it had the structure of a mite. And here it is. It's called Emping, and it's 430 base pairs in length. So the problem, though, is when you do computational analysis and you identify something that looks like it should be active, you've got to go back to the bench and prove that it's active. This is what we call a candidate. It's a candidate active element. You've got to then do an experiment that validates, that shows it moving around. So what Ning did was she took cells that were in the freezer for 4 years. She popped these cells out, and we had cell culture. So cell culture is an environment where DNA, where transposable elements have been shown to move around in other situations. So what we had is we had the DNA from the plant two plants, one before cell culture and then after cell culture. And the question we're asking is, can we see the movement of the Emping element? And we use a technique called transpose on display. This is what we used years ago. Now all you have to do is sequence a genome, and we'll talk about that later. But this was the technology available to us at the time. So what you do is you make a primer, and here we made a primer that was near the end of the Emping element. And then what we're going to do is we're going to take genomic DNA, we're going to cut it up with a restriction enzyme, and we're going to put adapters at the end of the, the genomic DNA fragments. We're then going to do PCR using the primer from the end of the genomic fragments and a primer from the Emping element. And you resolve this on a gel, and that's what you see here. So what you see in lanes 1 and 2 is a situation, it's a rice plant in 1, the DNA from a rice plant before cell culture. In 2 is the DNA from the cell culture. And all the bands that and nothing's happening. You see 1 and 2, they look exactly the same. That's because the Emping element is not moving around in that cell culture. Each of the bands comes from, at one end of the band is the Emping primer that you see over here. At the other end of the band is the region in the genomic DNA where the adapter sequence is. Okay? This is a modification of a technique called RFLP. I'm sorry, AFLP. So anyway, lanes 3 and 4 are far more interesting. And this is something that, there are, science is really slow, but there are these days you have, which you always remember. And this was a day, it was a Sunday morning, I turned on my computer, and Ning had sent me this picture that showed that the Emping element was moving around in the rice genome. And it was, needless to say, it made my day. So what you see in lane 3 is the plant before it went into cell culture. And there's only a couple of copies of Emping in that strain. However, after cell culture, there's hundreds of copies. So what we can actually do is cut out those bands, re-amplify them and sequence them and determine the position of insertion of Emping in the cell culture DNA. So what I want to show you here is where Emping fits in. Emping is a MITE. It is a non-autonomous element, as are all mites. It doesn't code for anything. It's only 430 base pairs in length. Shouyu Zhang, who was a graduate student in my lab, took the Emping sequence and blasted it, compared it to the entire rice genome sequence. And he found a single transposon that looked like it was the autonomous element for this family. So that is called Ping. So we have Ping, we have Emping, and there was only a single copy of that element in the entire Nippon Bare genome. And we went on over the years to show that the transposase from Ping is able to move Emping, and I'm not going to talk about that in this talk. Okay. So what I want to tell you about is the copy number of Emping, because I've told you that mites are great because they attain these really high copy numbers of hundreds or thousands, and yet I'm bragging about some element that has 50 copies. That's not a whole lot. And in fact, when we look at a lot of strains, what I've shown here is the Nippon Bare, the sequence genome, which has 51 copies as NB. And then when we look at a lot of other Japonica strains, and this was done in collaboration with Susan McCooch, we obtained this strain collection, we see that there are very low numbers of Emping, from 25 up to 38. That's not the burst that I talked about, which is... So fortuitously, two other groups in Japan had identified the Emping element, but they identified it in plants, in living plants. And I'm going to talk about those plants in a second, but when we analyzed those plants, we found that these 4 related land races, as is shown here, had over 600 copies of the Emping element. So this really said to us that here is an element that is capable and has increased its copy number very, very quickly. But the next question, remember, one of the things we're interested in is how can the element do this? Here were 4 strains where the Emping element had increased tremendously to 600 copies. The question, what I want to show you in the next slide, is just how closely related all of these strains are, how the major difference between these strains is the Emping insertion sites. And so what we can do, and this is again another transposon display, where in the first lane, the NB is Nippon Bare with its sequenced genome, and where there are 50 copies of Emping. Those are fewer than 50 copies, and I'm not going to go into why that is. It's the way the experiment is done. The next 3 lanes are 3 of the 4 land races where the Emping element, where there's 600 copies of the Emping element. And what you'll notice, first of all, is that the patterns are very, very different. So the insertion sites of the elements are very, very different in each of those. So from that, we concluded that the Emping element had, at least at some stages, amplified independently. These strains, at some point, there probably was one strain in which the Emping element had amplified, had activated, had become active. And then land races are strains of rice that farmers have grown in particular areas. So these strains then were grown in particular areas. They were kept separate. And now we're bringing them back together in the lab. So what you see is the different patterns, but you can see that there's many more bands. So we say that they burst independently in these 3 strains. What I want to show you in the next slide is just, not the next slide, but the next experiment, is just how similar these strains all are. So what you see here is using, in the first, the gel on the left, we've used the Emping element, the primer from the Emping element. Remember I showed you that before, in PCR. What we're using in this second panel is a primer from a different element. So exactly the same genomic DNA preparations. But the difference is one of the primers in PCR. This is a primer from a retrotransposon called Dashang, which was also identified in my lab. There are about 1,500 copies of this in the genome. But what you see, again, using exactly the same DNA preparations, is the pattern for all those strains are exactly the same. So what that, well, virtually the same. What that says is the major difference between these strains is the different insertion sites of Emping. Okay. Now what I want to tell you is I want to show you the experiment we devised, and this is Eunyoung Cho in my lab, devised this experiment to see if the Emping element was still transposing in these high copy number strains. And this is people in this area of transposon biology, people will look at one organism and see transposons and look at another and see them in different places, and they'll say, okay, the transposon is moving. They're probably not. What they're seeing is just polymorphism. The only way you can really see transposition, you can say something is transposing, is if you see it right before your eyes. And that's what I'm going to show you right here. So what's done is we have one plant that was grown up, one rice plant. And from that plant, Eunyoung took 10 seeds. Rice is a selfer. It self-pollinates. So all those seeds are virtually identical. She planted the seeds, she grew them up, she isolated genomic DNA. She did transposon display using the Emping primer, and what you see are the 10 lanes, the 10 individual plants there. And you'll see differences, okay? You'll see differences, okay? And I'll explain to that in a minute. So she took the last plant here, the very last one. She took 10 seed from that plant, and she grew the next generation. And she did the same thing, transposon display. She took seed from the last plant here, 10 seed from that plant, she grew it up, here's the last generation. So by comparing these 3 panels, and by looking at the white arrows, the white empty arrows, what you'll notice is that there may be, there's a band, say in the F1, that then in the next generation is segregating. Okay? So you see that band. That is a heritable insertion of Emping that is now segregating because when it inserts in the F1 generation, it's heterozygous, and in the next generation, we see it segregating. So essentially, by looking at this, what you can see is what I think is pretty remarkable. That is this rapid increase in the Emping copy number over a very, very short period of time, a couple of generations. And so we can actually, just by simply counting bands, we can determine how many new insertions there are per generation. And that's what I'll show you here. So we have basically found that there are approximately 40 new insertions per plant per generation, and that 80% of these insertions are heritable. This blew us away. We had no idea that a transposable element could increase its copy number so rapidly. So now we have the material to address the questions that one of the questions that I posed at the very beginning. is that how do transposable elements amplify without killing their host? And then the second question I will end the talk with is, does amplification actually benefit the host? So the first one is, so the way we're addressing this first question is, where are the elements going? Where are they in the genome? So the way experimentally, the way we addressed that question was to take genomic DNA from these plants and essentially amplify out the end of the element, much like in transpose on display, but we're not going to run a gel. So we're taking genomic DNA, and you'll see in a second, we're not taking it from one plant, we're taking it from a small population. We're using the primer from the Emping element and a flanking primer in an adapter. We're amplifying up all of these flanking regions. And then instead of running it on a gel, because we don't want to look at a few insertions, we want to look at all the insertions, we use high throughput sequencing. In this case, we used 454 sequencing to sequence tens of thousands of up to hundreds of thousands of flanking regions flanking the Emping insertion site to try to find out where Emping is. But more importantly, and this was an experiment done by Ken Nato when he was a postdoc in my lab, what he did is he figured out that the capacity for sequencing is so great that we don't have to restrict ourselves to one plant. He can actually determine where Emping is in a small population of plants, and he chose the number 24. So we took 24 rice plants, and he was able to barcode the PCR reaction so we could tell which plant the PCR products came from. And so what he did, so what you see on the left in the gel is you'll see, for example, some of the PCR products from from a subset of the 24 plants. And you could see that in blue, I'm showing you the bands that are shared by all the plants. In pink, I'm showing you the bands that are only present in one plant. The ability to look independently and at shared and unshared insertions is very powerful. Because what we want to know is not just what are the insertions that are present in all 24 plants. Okay? We want to know, and those we'll call old insertions. We want to know what are the new insertions, what are the ones that are just happening. And the reason is it's possible that the old insertions have been filtered by selection. So that over generations, insertions that are in places in exons or whatever have been removed because they've been detrimental. So by looking at shared and unshared, we're able to get the whole spectrum of new and old insertions. Old is not really old. These are insertions that have happened. This initial burst really happened maybe over the last 50 to 75 years. But these shared insertions unshared insertions have happened, in our greenhouse. So essentially, what he was able to sequence. So we know that if all of the barcoded plants or some of them show the same insertion, we call that shared. If only one of the plants shows an insertion, we call that unshared. So he was able to determine 928 shared insertion sites and 736 unshared insertion sites. Okay? And as I said before, these unshared insertions are de novo insertions. They just happened, and they're heterozygous. Heterozygous because when an insertion happens, it goes into one of the two alleles. And heterozygous is important because if it's a detrimental insertion, if it's a detrimental mutation, it's a recessive. It's likely to be a recessive mutation. So this is I don't expect you to see this. It's just really to impress you. What you're seeing is each of these graphs is a different rice chromosome. And the blue, I'm going to blow it up in a second, the shared insertions are shown in blue, and the unshared insertions are shown in red, and essentially shows that the Emping insertions are all throughout the 12 chromosomes of rice, so it's on every single chromosome. And this shows a single chromosome, chromosome 4, and we're looking at the insertions on chromosome 4. And chromosome 4 has a very large region of heterochromatin. And most of the transposable elements, the DNA transposons, don't insert into heterochromatin. They insert near genes, which are euchromatin. But we do have a few insertions that are in or near heterochromatin. So what we know, what we've learned from this analysis, first of all, the distribution of shared and unshared insertions is the same, is exactly the same. There is no difference in the insertion preference of either class. Remember I said before that mites prefer to insert into single copy regions, into genic regions. And sure enough, 91% of the insertions that we found were in single copy sequences. The genome average of single copy sequences is about 54%. So this is saying that mites do prefer to insert into single copy sequences. And that we found that even when it does insert into heterochromatin, that's actually inserting near genes that are in the heterochromatin. So now we've looked at a growth scale throughout the chromosomes. Let's look more closely and see where is Emping inserting in and around genes. So here's a summary with a very surprising result. So what we see, we're looking at insertions that are in the 5' untranslated region, in exon sequences, in intron sequences, and in 3' UTR. And we're looking at, again, a summary of a very large number of genes, and what you're seeing is the percentage of insertions. And so what we find is that the gray here is the expected number, is the expected number of insertions given the composition of the genome. The pink are the unshared insertions, and the blue are the shared insertions. And the only thing that really stands out on this histogram is this. We find that there are far fewer insertions into exon sequences than we would expect by chance. Almost 10-fold fewer insertions, both the shared and the unshared. And the unshared is significant, again, because it tells us that Emping prefers not to insert into exon sequences. How it does this, how it knows exon from intron, we can only speculate at this point, and I'll speculate a little later. So Emping has another insertion preference. So here is genic regions. That's what you're seeing at the top is around the gene. And what we're seeing, the x-axis, is the percentage of insertions. And the blue or purple bar is the actual Emping insertions, and the gray dotted line is our control. So if we just sampled genome sequences, what would we expect to see in those regions? And what we're seeing is where the dotted line is the transcriptional start site, and we go upstream from there to -1, -3, and that's in KB. And what you see is that there's a spike. So there's a preference for Emping inserting within 1 KB of the transcription start site. And when we take this together, you say, well, how can this possibly happen? We don't know, and that's obviously an area of intensive interest. One of the ideas is that in plant genomes and other genomes, it's known that the region just upstream of the transcription start site has fewer nucleosomes, as do exons. Exons have fewer nucleosomes. I'm sorry. Exons have more nucleosomes, so introns have fewer nucleosomes compared to exons. So it seems that the Emping is avoiding insertion into dense chromatin regions, or relatively dense chromatin regions. But this is pure speculation at this point. It's the only thing that's consistent with this pattern of insertion, that is avoiding exons and preference for insertion near the transcription start site. So let me summarize at this point, and that is we find that 91% or so of the Emping insertions are in single copy regions near genes, that exon insertions are 10-fold underrepresented, so the element is avoiding insertion into exons, and that insertions within 1 KB of the transcription start site are also enriched. Okay? And finally, that the distribution of shared and unshared insertions are indistinguishable, meaning that this is the insertion preference of the Emping element. I said we don't understand it, but this is the preference. So at the beginning of this section, I posed 2 questions that we wanted to answer with this series of experiments. The first one is, how do transposable elements amplify without killing their host? And the answer to that question for Emping is the following, and that is the rapid amplification of a successful element, and Emping is a successful element, has really a more modest impact on the host than previously thought. So this is actually, when I first saw this data, I was kind of disappointed because I was like, boy, it's just not doing a whole lot. And then it kind of dawned on me that that's what successful elements have to do. I mean, they in order to be successful, and success again is defined as being able to attain very, very high copy number, it has to do little harm. So the second question that is more difficult to address is, does the amplification actually benefit the host? And we have some data that suggests that it does, and it's experiments that we're pursuing now, and I'm going to tell you what those results are. And really, what we want to do is we want to look at the impact of Emping insertions on host transcription. So I showed you at the beginning of this talk the situation that we wanted in order to, the experimental material we needed in order to address the question of what is the impact of insertion on diversifying gene expression, for example. And what we needed were alleles that differ by the presence or absence of a transposable element. Now we have lots of those examples. So, as I said, we wanted alleles that only differ by the presence or absence of the MITE. And this summarizes really what we have now. We have 710 genes. That and EG4, I didn't show this before, EG4 is one of the land races. It's a strain that we determine the Emping insertion sites. So by comparing the Emping insertions in EG4 with the same genes in Nippon-Bari, we now have essentially 710 genes that have alleles that differ largely by the presence of the Emping element. Of those 710 genes, almost 400 have insertions within the promoter region, the 5' the promoter region. 120 have insertions or so within the gene, and 193 have insertions downstream of the gene. So the question we're asking is, what is the impact of insertion on transcription? To do that, Ken Nato, when he was a postdoc in the lab, compared the transcription of the 710 alleles in EG4 versus Nippon-Bari initially under normal growth conditions in the greenhouse. So to do this, he used a microarray of rice genes. He did microarray analysis of 31,000 plus rice genes. He isolated RNA from Nippon-Bari and EG4 seedlings. And essentially, he determined that for a significant percentage of these alleles, there was no difference in gene transcription. So for 78%, the transcript levels for these genes were the same in Nippon-Bari as they were largely the same in EG4. So this is a pretty benign effect on host transcription. So what you see in this slide is a comparison of the expression of the remaining alleles, that is those where we did see a difference between the transcription in EG4 and Nippon-Bari. And for approximately three quarters of those alleles, we saw upregulation in EG4. That is, the presence of the Emping element was correlated with increased transcription of the gene. And most of that difference, well, what you see here is most of that difference were insertions that were in the 5' upstream region. So this is upstream of the transcription start site. We do see also differences in insertions in introns, that many of the intron insertions, many meaning of the remaining 25% that show an effect, most of those, in fact, were upregulated. There weren't many that were downregulated except the few that are an exon, and that's understandable. So what I want to do now is to show you how we confirm this microarray analysis. So what you'll see, I'll just take one of these over here to the left. What you see is a particular allele, and this is OS, some long number. The insertion of Emping is at minus 2497. So it's 2.5 kb upstream from the transcription start site. And yellow here is transcription in EG4. Gray is transcription in Nippon-Bari. So what we're saying, what we're doing here is we're isolating RNA, and instead of using the microarrays, we're doing PCR, quantitative PCR. And what we find in every case we check, the EG4 allele, for the particular alleles we're looking at, the ones that showed upregulation by microarrays, we're able to confirm that result using quantitative PCR, that yes, indeed, there is more transcription from the EG4 alleles. Now, we have a problem, and this, I'll try to make this as simple as possible. There's another difference between Nippon-Bari alleles and EG4 alleles, besides the presence or absence of Emping. And that is that the allele in Nippon-Bari, which doesn't have the transposon in it, is in a genome that only has 50 Empings. Whereas the EG4 alleles, all of them, are in genomes that have, I show here 1,000, but 500 to 1,000 Emping elements. So it's possible that the differences that we're seeing in transcription between EG4 and Nippon-Bari is due to that load of 1,000 elements in the background. What we need is a control. We need a control where we can compare the alleles with and without Emping in the same type of genomic background. And again, I wouldn't be telling you that we need this control if we didn't have one. And so I mentioned at the beginning that EG4 is one of a couple of land races that have, in which the Emping element has burst, where we have many copies of Emping. And recall, I showed this transposon display, and so what I'm showing here is EG4 is one of these land races, another is A123, and another is A157. So, and the other thing you'll notice is that the Emping insertions in those strains, just by looking at the patterns, you could see the patterns on the transposon display differ. This means that the insertions are different. They're in different places in the genome. So this allows us to have valid control. So here we see alleles that differ between Nippon-Bari and EG4. The control is we can identify in, for example, A123, we can identify A1 and 23 that has the Nippon-Bari allele in it. Okay? So in that way, we're able to compare A123 and Nippon-Bari to EG4, and that way we sort of eliminate the complication of the 1,000 Emping insertions in the background. And I'll show you that data now. So here we have, what I'm showing is a histogram. This is a particular gene, OS01G. It's a rice gene, an annotated rice gene. The minus 600 means that it has an insertion of Emping 600 base pairs upstream from the transcription start site. And that's shown in this schematic below here. So what you see is that in Nippon-Bari, which is the gray, we see a level of transcription which is set arbitrarily as 1. In EG4, we see about 5-fold more transcripts. Now we also have to compare, we have the blue. The blue is the A123, which is another land race where there is no Emping in that position. So A123 has the Nippon-Bari allele, and despite having that allele in that background, no, not despite, even with the 1,000 copies of Emping in the background, we still see reduced expression of the gene. Okay, so the alleles, Nippon-Bari allele and A123 allele, we're getting about the same level of transcription. A154 has the same insertion as EG4, and as you see, we get increased transcription. So this clearly tells us that the difference in transcription is due to the Emping, somehow, we don't know how, is due to the insertion of Emping at minus 600 from the transcription start site of this gene. Here's another experiment. I'm not going to show you all of them, don't worry. Here's another gene. It's OS01G0 whatever. This insertion is at minus 2.5 kb from the transcription start site. Again, so what we have here in Nippon-Bari, the negative next to Nippon-Bari means that there is no Emping in that gene. The plus means there is. So here, EG4 and A123 have that allele with Emping. A157 doesn't. Again, the expression has to do with the presence of Emping, in this case, 2.5 kb upstream from the transcription start site. So, just to summarize this part, the impact of Emping insertion on nearby gene transcription, the vast majority of alleles we see no impact, no effect. This would be a neutral mutation, okay? Of the 710 alleles we're comparing, 111 we see upregulation of the nearby gene, and for 45 we see downregulation of the nearby gene. Okay. Now the question that we're going to ask is, does the presence of Emping affect transcription in a different way? Does it, in this case, confer stress inducibility on nearby genes? Now remember from McClintock's scenario, she mentioned the possibility that transposable elements are induced by stress. So here we're going to look at something a little different. We're going to ask, does the presence of a transposable element cause the nearby gene to be stress inducible? So this experiment, I'll lead you through this here, is we're looking at 3 different stresses: cold, high salt, and desiccation, dryness. So this is a gene which has an Emping element at minus 55. So 55 base pairs from the transcription start of this gene. And this is a gene that you'll see the control under normal conditions. It's one of those vast majority, 78%, that show no effect under normal growth conditions. That's what you see under control there. However, when we subject these plants to cold, and we meaning Ken Nato again, cold or salt, we see that the strain EG4, which has Emping at minus 55, we see increased transcription, okay, not much, but we see reproducible increased transcription, whereas the other high copy strains that don't have this allele with Emping do not respond, okay? So here's another example. This is an Emping element that in gene OS02, it has an insertion at minus 41 base pairs upstream of the transcription start. What we see is the alleles, here EG4 in blue and A123 in yellow have the Emping containing allele, and you see those are the ones that are induced by cold and salt. What's nice here is we're not seeing any effect of desiccation. We see a consistent effect of cold and salt. We don't know the mechanism for this. It is under investigation. So then the question is how, so one of the things you might wonder is, how is the transposon affecting transcription? Is it acting as an enhancer, or is it acting as a site, a new promoter, a site of transcription, initiation? So we have several intron insertions, and we can do the same experiment. Here we have OS, another gene, which has an Emping element only in EG4 in an intron. And when we do the same experiment, we see that under room temperature, RT is room temperature, normal conditions, there's no different in the transcription of the allele with and without Emping. However, when we look at the situation in the cold, we see that it's cold inducible. So here the transposon in a distant intron is affecting the inducibility of this gene. And we see that in the next slide also. This is another gene with a very large gene with an Emping element in an intron. And these introns are in the EG4 allele. I'm sorry, these Emping insertions are in the EG4 allele and in the A123. And again, we see that those two are inducible, suggesting that Emping sequences are in some way acting to enhance transcription under cold conditions. We didn't do this experiment we didn't test dry and salt. Okay, so let me give you conclusions from this part of the talk. The first thing is that we found surprisingly, or maybe we were surprised, but that's why you do experiments, to get surprised, and then you see the results and you say, oh, that makes sense, that massive amplification is largely benign. And when I say up to a point, we've caught this element in the act of amplifying. Obviously, at some point, if the number of elements transposing gets too great, it's going to start causing some damage. And that's one of the things we're really, really interested in. When does this activation stop? What happens? And we don't know yet. That the amplification has a subtle impact on the expression of many genes. It causes stress induction, it induces the expression of some genes, but it's really tweaking them. Most of the expression we see is maybe a 2-fold, 3-fold increase. And again, it produces stress-inducible networks. And I say cold and salt. Others, I'll give you a few tastes of where this is going. And the other thing that's significant is that it generates dominant alleles. So if you think about a population, remember I said that when these elements insert, they're heterozygous. That if it caused a phenotypic change, that overexpression will be a phenotypic change that can be seen possibly in a heterozygous organism. So we don't have to wait for this to become homozygous. So I want to go back to McClintock's scenario again, and that transposable elements, her scenario for how transposable elements can function as tools to generate diversity. Transposable elements usually don't move around, and we know that now. We know that the vast majority of transposons in the genome are inactive, even though the genome is, genomes are 50 to 80%, 20 to 50 to 80% derived from transposable elements, that most are inactive, that they are inactive because they've accumulated mutations, or the few that are active are being epigenetically restrained by the host. That it's possible that somehow stress conditions may activate transposons. Now, I haven't shown you that. We started with a strain Emping, the EG4 strain, where the system was active already. We don't know how it became active. That obviously is something that we're very interested in. And we think that it's possible because EG4 and Emping are present in most rice strains, that in most rice strains these elements are epigenetically silenced, but that somehow in these few strains, these land races in EG4, that the element became activated. We do not know how. That obviously is an area of future research, and that's a critical area because that's how we think most genomes are sort of poised, many genomes. They have the ability for active transposable elements to start amplifying, but how that switches, what is the switch and how it's thrown is a subject of future research. Again, that the movement of transposable elements generates genetic diversity, increases the mutation frequency. McClintock looked at mutagens. She looked at elements that geneticists look at mutants. These are, as I said at the beginning, these are not insertions that will benefit the organism. However, we've been able to identify an element where most of the insertions are benign. And that, as we said, a rare TE-induced mutation may be adaptive. So I want to sort of speculate a little bit and tell you about how we sort of fit Emping into this model, that somehow stress could have induced ping. Ping is the autonomous element. And again, this is a black box. We do not know. We weren't there when this happened. We came upon the strain, or our Japanese collaborators came upon the strain when it was already active. This leads to the massive and rapid amplification of Emping. That we're seeing, it's still in progress. This generates tens of thousands of new alleles. Now, we looked at 24 plants, but imagine a field of 1,000 plants. Emping accumulating 25 to 40 new insertions per plant. What's really interesting, the point I haven't made, is rice is a selfer. So it selves. There is no new genetic information coming into populations. The same genetic information is being scrambled up by recombination or whatever. Emping is a way, transposons are a way to dramatically diversify the genetic material without introducing without having gene flow into the population. So this, what we're hypothesizing, we see it at the transcription level, that this amplification creates transcriptional changes, and we're hypothesizing that these changes can lead to quantitative variation, and that so changes in cold tolerance, changes in drought tolerance, changes in desiccation. But that's the point that we're now testing. And I'm going to end by telling you about, very briefly, about the experiments that we're currently doing to address the question, really the smoking gun question. And that is, what is the phenotypic consequences of the Emping burst on EG4? Does this, we've talked about transcriptional changes, but are there phenotypic changes that go along with those? And the way we're doing that is in a number of ways. The first thing, and again, we're taking advantage of the wonderful new high throughput sequencing technology. So one of the things that's allowed this project to move forward, and I think for most of us in molecular biology, it's the technology that really drives the questions that we ask, and that we can get deeper and deeper into a particular problem as the technology changes. And the availability of high throughput sequencing has allowed us to address questions that we didn't even dream about, as recently as 5 or 6 years ago. In this case, what we can do is, we've shown here, we can, well, first of all, we know that Nippon-Bari and EG4 differ phenotypically in several characteristics. They're different in flowering time, they differ in average height, they differ in some of the stress responses. We want to know, for example, if any of those differences are due to one or more Emping insertion. In order to figure this out, the first thing we have to do is to figure out we have to know, is there more going on in EG4 in those land races than just Emping amplifying? Because remember when we sequenced the insertion sites, we did an approach where we used PCR primers and only amplified the element and flanking sequences. Well, again, we were limited before by the technology. Now we can actually sequence the entire genome, and in fact, we've done that. So EG4 is currently being resequenced using next generation technology so that we can see, is the Emping amplification the only thing, the only transposon that's amplifying in the genome? And so far, the preliminary answer to that is yes. It appears that Emping is the only transposon that's amplifying at this time. The other thing is we're doing transcriptomics. Rather than looking at particular individual genes, we're looking in a strand-specific way at the entire genome of Emping, EG4 and Nippon-Bari. And this is done in collaboration with Tom Brutnell's group at Cornell, where they've developed a really nice protocol to look at single-strand RNA-seq. So now, how do you, the way that's traditionally used to find the regions of the genome that are responsible for quantitative traits is a mapping population, a recombinant inbred population. And our collaborators at Kyoto University in Tanasaka and Okamoto's lab have over the last decade been developing this incredibly valuable resource. So what they did over 10 years ago was to cross EG4 with Nippon-Bari. Now what I want to point out is these are inbred lines. So all of they have 2 copies of exactly the same gene at every single locus. So we have EG4 crossed with Nippon-Bari. We have our F1 progeny, many, many F1s. Those F1s are then self-crossed for 10 generations. So we now have growing in this country 275 recombinant inbred lines. These lines have mosaic chromosomes that are derived from EG4 or Nippon-Bari, and they are displaying different traits. So we're phenotyping them now, and I'll talk about that in a second. So the question really, and I think on the next slide I go into more here. So we're looking at these recombinant inbred lines. We're assaying RILs for recombinant inbred lines for morphological traits and stress responses. And we're doing something that, again, I would never have thought would be possible, even in the grant application I wrote a couple of years ago. I didn't even write that we would do this because it wasn't affordable, but now, again, the technology, the costs have come down. We're actually resequencing all 275 RILs to find out exactly the mosaic structure of their chromosomes, what part came from Nippon-Bari with its Empings, what part came from EG4 with its Empings, so that we can ultimately correlate the Emping insertions with the various phenotypes. Now again, this is just correlative at this point. We then will have to prove that the candidates that we find, the Emping insertion alleles, are the ones responsible for that phenotypic difference. So many of us, and many of us in the field, think of Barbara McClintock as the first genomicist, the first person who thought of the genome as an entity, not just as single genes. And there's a quote from her Nobel lecture, which I want to end with. And it's her thinking about the genome, and it really presents a challenge that I have at least felt and have gone with my lab. And that is, in the future, attention undoubtedly will be centered on the genome, with greater appreciation of its significance as a highly sensitive organ of the cell that monitors genome activities and corrects common errors, senses unusual and unexpected events, and responds to them, often by restructuring the genome. She ends by saying, we know about the components of genomes that could be made available for such restructuring, in part the transposable elements that she discovered. We know nothing, however, about how the cell senses danger and instigates responses to it that are truly remarkable. I'd like to say that we're beginning to understand that black box of the connection between the outside world and the genomic changes, and I think transposable elements are certainly part of that.